Welcome to AP Biology. This video is an overview of DNA structure and replication. First we look at the structure of DNA. DNA is a polymer. It stands for deoxyribonucleic acid. You can see based upon this picture here that DNA is double-stranded. Here you see one strand of DNA. Here you see the other strand. The monomers that make up DNA are called nucleotides. Here we see one nucleotide, and a nucleotide is made up of a phosphate group, a sugar, which is going to be deoxyribose, and a nitrogenous base. There are 4 different nitrogenous bases that can make up DNA, so there are 4 different types of nucleotides. You have a nucleotide that has a T, that stands for thymine. You have a nucleotide that has an A, that has adenine. You have a nucleotide that has a G for guanine, and you have a nucleotide that has a C for cytosine. If you look, A and G are both purines. Purines have a double ring structure. C and T are both pyrimidines. They have a single ring structure. So DNA is made up of a long string of nucleotides that are bonded together. DNA is double-stranded, so there's actually 2 long strings of nucleotides, and the 2 strands are held together in the middle by the hydrogen bonds. If you look at the structure of DNA, A is always bonded with T, C is always bonded with G. If we look at the DNA, it has a ladder structure to it, where the phosphates and the sugars, they make up the sides of the ladder. We call it the phosphate sugar backbone. So here you have one side, here you have the other side, and the nitrogenous bases make up the rungs in the ladder. And when we read the message in DNA, it's really the sequence of the nitrogenous bases that matter. The phosphate sugar backbone never really changes. Now, if you look at this DNA that we see here, it's anti-parallel. To be anti-parallel means that the 2 strands are running in opposite directions. One is running this direction, and one is running in the other direction. We call this anti-parallel. This side we call the 5' side, and this side we call the 3' side. So then that means that this would be the 3' side, this would be the 5' side. So one's running 5 to 3, one's running 3 to 5. This is going to play an important role when we look at the process of DNA replication. This DNA ladder that's anti-parallel gets twisted around and it forms a DNA double helix. Here we see a list of experiments that were performed that proved various aspects of DNA. The first set of 2 experiments here, the Griffith and Avery experiment and the Hershey and Chase experiment, they both proved that DNA was the genetic material and proteins were not. The Griffith and Avery experiment was the first experiment performed. Griffith performed a process called transformation. During his transformation experiment, he injected mice with different strains of bacteria, and what he found is that the genetic information traveled from one bacteria to the other bacteria and transformed that bacteria into a harmful bacteria. Avery extracted the material that was being transferred and discovered that it was DNA. So it was really the first experiment that showed that DNA was the genetic material. And the key word to link with the Griffith and Avery experiment is the term transformation. The next experiment was performed by Hershey and Chase, and this is the experiment that really confirmed and clinched the idea that DNA was the genetic material. And they did an experiment using bacteriophages. Bacteriophages are viruses that infect bacteria. We know that a virus is made up of 2 materials. It's made up of proteins and it's made up of DNA. And Hershey and Chase wanted to see which one of those two the virus was inserting into the bacteria. They labeled one group of viruses with radioactive phosphorus for the DNA, and they labeled another group of viruses with radioactive sulfur for the proteins. What they found with their experiment is that DNA was the genetic information being inserted into the bacteria, and thus DNA really is the genetic information. The next set of experiments deals with the structure of DNA. Maurice Wilkins and Rosalind Franklin, especially Rosalind Franklin, she performed X-ray crystallography, and using this procedure she was able to make a picture of DNA, or form a picture of DNA. Two other scientists named Watson and Crick analyzed her picture and discovered that DNA was a double helix. So Watson and Crick were credited for the discovery of the structure of DNA being a double helix. They used Rosalind Franklin's data in order to make their discovery. So both Maurice Wilkins and Rosalind Franklin and Watson and Crick all performed, or all had an integral part on discovering the structure of DNA. Erwin Chargaff, he performed an experiment and he discovered that the percent of adenine in an organism equals the percent of thymine, the percent of cytosine equals the percent of guanine, and therefore A is bonded to T and C is bonded to G. So if an organism has 30% adenine, that means that they also have 30% thymine, which means that they have to have 20% cytosine and 20% guanine as well. Messelson and Stahl, they discovered that DNA replication is semi-conservative. We'll be seeing what that means in a second. Beadle and Tatum discovered the one gene, one polypeptide hypothesis. They discovered that each gene is the blueprint for the creation of one protein within the body. DNA replication. The important key term to link with DNA replication is semi-conservative. We know that DNA is double-stranded. The original DNA molecule we call the parental molecule, and it's made up of two parental strands that you see right here. During DNA replication, the two parental strands are going to separate and two new strands are going to be built. So if you look at each new DNA molecule, which we call a daughter molecule, each daughter molecule is going to be made up of one old parental strand and one new strand. So in a way, DNA is never lost. DNA never dies. All the old DNA gets incorporated into the new DNA, with each new DNA molecule containing half of the old and half new. We refer to this as semi-conservative replication, and it was proved by an experiment performed by Messelson and Stahl. Here we see the Messelson and Stahl experiment. They incubated bacteria with an isotope of heavy nitrogen 15. So all the DNA that this bacteria made contained the heavy isotope nitrogen 15. Here we see the two original parental strands of DNA, or one DNA molecule. They took the bacteria and they then incubated the bacteria that have the heavy nitrogen 15, and they placed it into a lighter isotope of nitrogen 14. By doing this, all the new DNA that was made is now going to contain the new nitrogen 14. So if we look at the original DNA, if the original DNA from this first group was put in a centrifuge, since it contained all heavy nitrogen 15, it would form a heavy lower band towards the bottom. When they placed the bacteria with a lighter isotope of nitrogen 14, Now all the new DNA that's made was made with nitrogen 14. You can see that each new molecule, each daughter molecule, is going to contain one old heavy nitrogen 15 strand and one new light nitrogen 14 strand. So when they put this DNA into the centrifuge, they got a medium hybrid band in the middle because it's a combination of 15 and 14. This was the first round of replication. They let the bacteria go through a second round of replication, also in the nitrogen 14. So again, all new DNA that was made would be made using the nitrogen 14. This nitrogen 15 never goes away. It's always incorporated into the new DNA. So this one goes right here, and the new one that was made is made using nitrogen 14. Here, this nitrogen 14 stays, and the new one that's made is also made with nitrogen 14. So after the second round of replication, they put the DNA into a centrifuge, and they got a medium hybrid band again, and that's because of this one right here, and they got a light band because of this one right here, contained 1414. So these experimental results that you got, or that they received after the second round of replication, they showed semi-conservative replication to be true. The DNA never died. Half of each old molecule got incorporated into the new molecule. So no matter how many more rounds of replication they let it go through, they're always going to receive this medium band because this nitrogen 15 is never going to go away, and they're going to keep receiving light bands because of the nitrogen 14. Here we see the actual steps of DNA replication. It's not important to know every single little detail. There are 5 major enzymes that are used. The first enzyme is called helicase. Helicase is going to unwind and open up the DNA. It creates a replication bubble that's made up of 2 replication forks. So here you see one fork here, here you see the other fork there, making one replication bubble. DNA replication is going to extend out at each fork. This is going to happen until all the replication bubbles converge. Helicase is also helped by a couple other enzymes. You have another enzyme called topoisomerase, and you have proteins called single-strand binding proteins. DNA replication begins with an enzyme called primase. Primase lays down what we call RNA primers. Primers are the site at which the new replication is going to begin. It's kind of a starting point, and it's made out of RNA. If we look at the 2 DNA strands, we can see here how they're anti-parallel. One's going 5' to 3', and the other one's going 3' to 5'. One of these strands is going to be called the leading strand. This one here is on the top. And one of them is going to be the lagging strand. The leading strand is going to receive 1 primer, and the lagging strand is going to receive multiple primers. We'll see why in a second. The enzyme that actually replicates the DNA is called polymerase III. Polymerase III reads the template strands of DNA, and it lays down the new nucleotides, but it only works in one direction. It only makes new DNA from 5' to 3'. Since it only makes the new DNA 5 to 3, it has to start at the 3' side of the template strand. So the leading strand, this one on the top here, is the one that goes nice and easily, where polymerase goes in the direction that it's supposed to go. The bottom strand, we have a problem. We want replication to extend this way, but polymerase can only go in the opposite direction. This is why we lay down multiple primers, and then once multiple primers are laid down, polymerase III is going to come in and extend each primer. and it's going to create a sequence of DNA fragments. These fragments that are created in the lagging strand are called Okazaki fragments. So the next step that happens is that another enzyme called polymerase 1 comes in. Polymerase 1 is going to remove the RNA primers, and it's going to replace the RNA primers with DNA. Finally, the last enzyme used is called ligase. Ligase is then going to come and connect all these Okazaki fragments together, making one continuous strand. If we look at the end result, we see it here on the slide with semi-conservative replication. We had two original parental strands that we call the template strands. They're going to separate. That's the job of helicase. Primase lays down primers. Polymerase 3 extends the primers. It's going to read the template strands, and it's going to lay down all the complementary nucleotides. The only problem is that polymerase 3 only goes in one direction. So one of the strands is made continuously, and the other strand is made in short fragments that we call Okazaki fragments. The end result is, though, that one parental molecule becomes two daughter molecules, and each daughter molecule is identical to each other. They're exact replicas. Each new molecule is made up of one old strand, and each molecule is made up of one new strand. This is why replication is referred to as semi-conservative. That completes this video.